Welcome to Applied Genetics, the lesson number 3. So we've talked about the lesson number 1 and 2. So what are examples of selective breeding? So we want to talk about selective breeding, test cross, and inbreeding. We've talked about hybrid as organism which is heterozygous for a specific trait. Let's say, for example, we talked about two cats. One is black with this, and the other one is white with this. So you want to cross these two cats, you're going to get the gamete for the black and the gamete for the white. Then you would cross them. Then that gives you heterozygous dominant cat. So this is going to be the F1 generation. So if this is a hybrid, F1 generation hybrid, you can decide to cross more time to get more hybrid from these heterozygous which you got. So that would give you this, 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 and this. So we still have 2, which is hybrid. So this is hybrid. So this one is it's called like the parent allele, like this. And this one is also called like parent allele. So this is going to be F2 generation. So the F1 generation hybrid is this, and this is the F2 generation hybrid, which is going to have 1 to ratio 2 to ratio 1. All right. So let's move on to the next one. So for thousands of years, humans have bred plants and animals for specific desired traits. When you desire a trait or you cherish a trait, you would want to get the trait by crossing with the organisms that have the trait. So the process by which desired traits of certain plants and animals are selected and passed on to the future generations is called selective breeding. So methods for selective breeding include hybridization and inbreeding. So hybridization is a process by which hybrids are being made, as I've showed you earlier. So inbreeding is allowing the organism of the same trait to breed with each other. For example, let me show you what inbreeding looks like. Inbreeding would look like this. Let's say we have this for black cat and this for white cat. So inbreeding will look like this. You breed this and this against this and this. So you're gonna have something like this. You're gonna have something like this. So if you breed as many times as possible, you would have the same alleles because it's inbreeding. So, and also if you decide to do inbreeding for this, you get something like this. So among the differences between inbreeding and hybridization is hybridization usually lead to heterozygous offspring, but inbreeding would most of the time give you a homozygous offspring. So now hybridization occurs by crossing parent organisms with different forms of traits to produce offspring with specific traits. So when you cross parent generation with different parent generations to produce offspring with a combination of the traits you actually admire, that's hybridization. So the process of creating hybrid is called hybridization. The disadvantages of hybridization is that it's really, really expensive and it takes a long time before you actually get the traits you want. So this is one of the disadvantages of hybridization. Advantages include adaptability. You would have organisms that would be able to adapt properly to the environment. And also it can lead to an increase in the number of offspring. So the number of offsprings which is going to be got will be so many because you have to do that lot of times before you can actually get the combination of the traits you need. Inbreeding, on the other hand, is a process by which two related organisms are bred. As I showed you before, you breed the same related organisms. So because there is a trait or there is a quality you actually desire, you admire. So, and you want to eliminate the ones you don't decide, the ones you don't desire, or the ones you don't admire. So in a future generation, you want to have a trait. You don't want to have organism with all of the traits in which you really want. Inbreeding is used to create a pure breed in horses, dogs, and other animals. The disadvantages of inbreeding is that it causes harmful recessive traits. So the harmful recessive traits which we have can be passed on to the next generation. So that is going to cause a lot of problems. Now let's talk about test cross. When you do hybridization, you don't actually know the genotype of an organism. So what you're going to do is you're going to make a test cross to be able to know, ascertain what genotype the organism is got. So breeders can determine the genotype of hybrid by doing a test cross. So what is a test cross? So a test cross involves breeding an organism that has an unknown genotype. So the organism in which you don't know the genotype, you want to breed it with the ones in which you know the genotype. So you want to breed the ones with the unknown genotype with the one in which the genotype is known, but mind you, you have to breed with the recessive alleles so that you can easily know if the dominant would actually come out. So if the parent's genotype is homozygous dominant, all of the offsprings will have to be dominant phenotype. So the meaning is, if you want to cross and that offspring happen to be homozygous dominant like this, and you're trying to cross it with because you have to, it's a process. You have to cross with a recessive for that same trait. So you're crossing with a recessive. So if the organism with the unknown genotype is homozygous dominant, so all the offspring will be 100% heterozygous dominant. So you have ratio one. You have one. You have one, or you have 100%. 100% means 100 over 100, and that gives you 1 over 1, which is 1. So that means if you have the parent is homozygous dominant, but if the parent is heterozygous dominant, that is to say, you have one uppercase and one lowercase. So what would happen is you would have these and these crossed against these and this. It's a must. You have to cross against the recessive allele. So what you're going to have is this. So you would have 50 here, and you have 50 here. So it's going to be ratio 50 to what? To 50, which is 1 to 1. So that's how you got this. So the genotype is going to be, um, so the genotype is going to be ratio 1, is going to be this, is going to be, let's say, for example, black. Let's say uppercase is black cat, and this is white cat. So this cat would be black, black, white, white. So you have two black cats with two white cats. So, and also if you want to do the genotype, you would see that the genotype is 2 to ratio 2. And also the phenotype is also what is also 50 to 50, which is 1 to 1. So this is the way it is if you have heterozygous um, if you have an heterozygous dominant for the unknown genotype. So how do you perform test cross? For example, we want to have a case study here. We have a grapefruit with a white color. So the white is dominant, is having uppercase W, and the red is recessive with a lowercase w. So the red grapefruit must be homozygous recessive. So this, the one you know, must be homozygous recessive. So the white, if the white is homozygous dominant, or it has to be either homozygous dominant or heterozygous dominant. So in a test cross, a tree with white fruit color or unknown genotype is crossed with a tree with red fruit this. So once you cross this is the result. So let's say you have homozygous dominant. So if the white grapefruit tree genotype is this, all of the fruit would have heterozygous what? Dominant white color. So all of them would be white, which is this heterozygous dominant. So let's say it's heterozygous white, and you try to cross it with a known red recessive allele. So what would you have? You would have, if the white grapefruit tree is this, so the fruit will be half of the resulting tree will be white and half will be red. So it's gonna be this ratio 2, this will be 2 ratio 2, this 1 ratio 1. So this is it. So which phenotypic ratio would result if a parent homozygous dominant for a trait is used in the test cross? So the question says, which phenotypic ratio will result if a parent is homozygous dominant? For homozygous dominant, it's going to be 1 because it's going to be 100% heterozygous dominant. So what is the genotypic ratio of the offspring in the test cross shown? So the genotypic ratio means the genotypes you see. What are the genotypes? We see this genotype here is the same as this. So that's 2. This one here is the same as here. Then ratio 1, ratio 2. So that gives you 1 ratio 1, ratio 1. So that's going to be B. So what is the advantage of inbreeding? Inbreeding is costly? No. It maintains pure breed? Yeah. Inbreeding is, what is the disadvantages? Okay, the disadvantages is it pass on harmful recessive trait. So it pass on harmful recessive traits. So this is the answer. Which term describe the processes in which people cross parent organisms with different forms of a trait to produce offspring with a specific trait? So when the process of crossing parent organisms with different forms is called hybridization. So this marks the end of lesson 3, and I wish you all the best in your